Aim: the comparative study of the rate of transpiration in the upper and lower surface of the leaf. Materials required: a broad-leaved potted plant, 5% cobalt chloride solution strip, slides, leaf clamps, desiccator. Desiccators are sealable enclosures that contains desiccants used for preventing moisture-sensitive items such as cobalt chloride paper for another use, and a common use of the desiccator is to protect from moisture. Filter paper, forceps, rubber bands, measuring cylinder, stopwatch, and cotton. Theory: transpiration is the loss of water in the form of water vapor through the stomata. The number of stomata present on both the surface of the leaf differ in most plants like dicots and monocot. The rate of transpiration from two surfaces of the leaf can be studied by comparing the loss of water vapor from the two surfaces, such as upper and lower epidermis, and that can be demonstrated by cobalt chloride paper test. Cobalt chloride CoCl2 is blue colored in anhydrous form, that's dry, but that turns to pink color when it comes in the contact with water. Time taken for cobalt chloride paper to change color is inversely proportional to the rate of transpiration. High rate means less time and slow rate means more time to turn to pink, and that also depends on the number of stomata on the leaf surface. Less time taken by the leaf to change color shows high rate of transpiration. That means there are more number of stomata and high rate of transpiration, while the more time taken is slow rate of transpiration because there are less number of the stomata. In mesophytic plant, the lower surface of the leaf has more stomata than the upper epidermis of the leaf. Usually, the rate of transpiration is measured by the potometer, which estimate water loss by weighing. Why transpiration? So transpiration is necessary for maintaining the temperature of the leaf and also useful in transportations of different materials to and from the various tissues of the plant. For example, transpiration pool theory. Procedure: preparations of cobalt chloride paper. Number 1, prepare 5% cobalt chloride solution. Then cut the filter paper stripes and dip them into CoCl2 solution, and then dry these stripes under the flame or under sunlight. This is ready-made cobalt chloride solution. Take 5 gram cobalt chloride and dissolve it into 100 ml of water. Then take the filter paper stripes which we cutted in this manner, then dip them into the CoCl2 solution, either prepared solution like this or the freshly prepared CoCl2 solution. Then let them soak. After some time, when it is sufficient soaked, then we can dry that stripes under the sunlight or under the flame. After some time, put the stripes under sunlight for drying or under the flame. On drying, the stripes are blue in color as you see. Take potted plant with the big leaf and start the practical. As you can see in this video, take a plant, potted plant with the broad leaf, then take 2 slides, put the dried CoCl2 stripe in proper arrangement, one on upper epidermis, one on lower epidermis as in this video you can see, and then clamp it. You can either use the rubber band or you can use these clamps. So yes, clamp it and then start observation. Now after some time you may see that the blue color of the stripes fades away and slowly that turns into pink color. The turning of pink color of the stripes shows that the leaf is releasing water through the stomata and transpiration is occurring. Within no time you may see that the stripe is turned to pink totally at the lower epidermis. You can see that means the lower epidermis has high rate of transpiration and more number of stomata than the upper epidermis. Over here upper epidermis also turns to pink, but after the lower epidermis. Okay, so the rate is at the lower epidermis high, upper epidermis is low. Now this is a second plant. You can see I am handling this stripes with the forceps and I am taking the help of other students. Yes, so like that, try. Okay, do not handle it with the bare hand. Your hand must be dry, and I am using this rubber band to put this stripe on this leaf. After this, wait for that so that this blue stripe will turn to pink, and we have to apply the Vaseline at this edges so that the moisture from the air cannot enter inside and you might not get the false result. Okay, now you can see. Now we have to wait. Take your watch or stopwatch and observe the time taken for the stripes to turn into pink from blue, and that time we are going to write as a result. Now you can see actually it is it has taken very long time, but still the stripes are not totally turned to pink. You can see little bit margin is only pink. Now this is one more plant with the broad leaf. In the same manner, try to put a new slide and handle it with the care. Do not touch with your bare hands. Be careful. If you have Vaseline, apply it. Then again the same way we have to follow the steps. Take the stopwatch and observe the time. This plant had taken almost approximately 3.5 minutes around, and where the time is seen on the screen, you can stop the video and observe the time taken. The lower surface is taking a lot less time than the upper epidermis. This shows upper epidermis is having less transpiration and lower epidermis is having high transpiration. Yes, you can see the result. This is turning to pink slowly. Yes, the video is little bit fast forwarded so that you can enjoy this very fast. We had tried many practicals, so you can also do the same way and try as many plant you can perform this practical. Write down the name of a plant, then write down the note down this time taken for this stripe to turn into pink, and then write that into the observation table. As in the observation table, you can see there is a name of plant, the upper epidermis, lower epidermis, and the time taken for change color blue to pink. For example, write the name of plant, then time taken in upper epidermis and time taken in the lower epidermis. Write down the time taken for color change on the upper surface and lower surface. The cobalt chloride paper turns pink on the lower surface after some time, but upper surface takes more time. Result: as the number of stomata are less on the upper epidermis of the leaf, hence the rate of transpiration is slow, while the number of stomatas are more on the lower surface, hence the rate of transpiration is higher.